¿Sí? Ana, ¿nos oyes?
Sí, os oigo.
Vale, pues buenos días a todos y bienvenidos a la segunda sesión de IMIM Seminars at Home. En primer lugar, nos gustaría agradeceros de nuevo la buena acogida que ha tenido la iniciativa. Ya hemos solucionado el problema de la limitación de solo 100 participantes, así que esperamos que a partir de ahora nadie tenga ningún tipo de problema para acceder a las sesiones. Y para los que no os conectasteis en la primera sesión, solo explicaros un poco que esta iniciativa es una idea de la nueva dirección del IMIM que persigue dos objetivos. La primera de ellas es alinearnos un poco en el tipo de investigación que se está haciendo en la institución, aprovechar este momento de menor actividad experimental para ello. Y el segundo es contribuir a las acciones de Open Science del IMIM. Esta sesión, igual que se hizo con la anterior, será colgada en el canal de YouTube del IMIM, de modo que todos podáis acceder a ella. Paso a darle la palabra al director del IMIM, Joaquín Arribas, quien quería hacer un par de comentarios sobre la ponente de la sesión de hoy, Ana Bigas. Joaquín, cuando quieras.
Muchas gracias, Vanessa. Quería empezar también, como Vanessa, por agradeceros la participación en estos seminarios. Creo que es muy importante que los científicos continuemos trabajando independientemente de las circunstancias. Sabéis por lo que estamos pasando, pero eso no nos hace pararnos y seguimos pensando en iniciativas para adaptarnos y seguir trabajando y seguir contribuyendo a que la ciencia progrese. Es para mí un placer especial hoy simplemente presentar a Ana, aunque la presentación ya la ha hecho Vanessa, porque aparte de ser, como todos sabéis, una de las científicas más valiosas del IMIM, estoy encantado de anunciar hoy que es la nueva directora científica de CyberOnc, lo cual creo que es una responsabilidad más que merecida en el caso de Ana, y además como director del IMIM para mí es un gran placer y creo que es un éxito para la institución que aumentemos en visibilidad y que esta responsabilidad haya caído a una persona que lleva tantos años trabajando tan bien y contribuyendo tanto al avance de la ciencia en el IMIM. Solo quería deciros eso, agradeceros de nuevo la asistencia, and now let's go for the science. Ana?
Okay, then the pleasure is mine to have this second seminar of IMIM at Home, and I really want to thank Joaquin and Vanessa for the opportunity to have the seminar today, which is a special day, obviously, even considering the situation, but trying to keep things going and obviously thinking about the future. I think this is the best that we can do right now at this moment is to think about the future. So I'm going to today I'm going to talk about one of the important lines of research that we have in the lab. We've been working for several years in deciphering the mechanisms of different processes that happen in our cells in a normal way, but we always focus in those that can help us to understand what happens when it goes wrong and when we have, for example, an oncogenic process. However, today it's going to be more on a normal side. I think this is a project that I have been working on in most of my life, because the idea is to understand how are hematopoietic stem cells regulated and how are these cells that maintain the blood in our systems, how can they be maintained for the whole time of the life of our lives. We know that the adult hematopoietic system, probably all of you know that this is like a hierarchical system that at the top, we have these hematopoietic stem cells that are responsible for generating all kinds of specialized blood cells and immune cells, and at the end, the cells that will be responsible for many functions in the organism, including fighting against something tan important as the infectious diseases. These hematopoietic stem cells, they reside in the adult life. They reside in the bone marrow. We know a lot about this. Scientists have been studying the stroma, the conditions in which these cells are maintained, and how these cells can handle these functions for the rest of the life. However, what I will be focusing today is something that is not so well known and studied, but it is very relevant to understand how a cell is specified and is generated. And in this case is the generation of hematopoietic stem cells, which, as much as we know right now, it takes place during embryonic life and in specific conditions that actually is preparing, we have developed in evolution to be able to generate this type of cells that are very robust and that they will have this incredible capacity that is so basic to maintain the life of an organism and to maintain a healthy organism. So we're going to be talking about the embryo. This is the studies that we are doing are mainly in the mouse embryo, and they are we'll be talking about this period of development, which goes more or less from it's the mid-gestation period where all the tissues are being formed in the embryonic life. The very briefly, what do we know, and what in the last probably, I would say 10-15 years, we have actually advanced quite a lot on how these cells are made, and we have changed our vision of how blood is made for the first time in the embryo to from a vision that was actually very simple in very early life, in which some hemogenic endothelium in the yolk sac, in the extraembryonic tissues, even before we have even before organogenesis takes place, there are some blood cells that appear in the yolk sac, in these extraembryonic tissues of the embryo. These blood cells were thought to be the first cells that would contain stem cells, and for many years scientists thought that these were the right cells to study if we wanted to know more about the stem cells. However, with the years, we have many contributions from many scientists have shown that this is not the case. The yolk sac, this very early time hematopoietic development is something like an emergency hematopoiesis that is thought to be used to help the embryo to get oxygen. Once the size of the embryo is too big to have the exchange of gases as it has been happening up to now. So once the first cells are made, and this mainly covers erythrocytes and macrophages, and this will be the responsible cells for doing the early functions that the embryo needs. Once these basic functions are covered for the embryo, another process starts that is much more complex, that takes place in the main vessels, in the main vessels, aortic vessels of the embryo, and it's known to be a process that will generate the hematopoietic stem cells. This happens in this area of the embryo, which is the aorta, and it's not happening all over the embryo. It's mainly restricted to where the tissues, the mesonephros and the gonad tissues are also developing. And for this is why it's known as an AGM tissue, because of the names of aorta, gonad, and mesonephros. It's called AGM, and in this tissue is where we can find and define the first hematopoietic stem cells, and I will tell in a minute what we mean by that. After these cells are generated, we know that the process doesn't finish here. These cells migrate to the liver. The liver becomes later on the most important hematopoietic tissue. And there is also a lot of things going on in the liver, with cells that will be amplified, and probably there will be the selection of the most robust hematopoietic stem cells that will then, close to birth, move to the bone marrow, and this is where they will stay for the rest of the life of the organism. So this is what it looks like. So these are the cartoons. In the cartoons, if we made a transversal section right here in the aorta of the embryo, we would be able to see the endothelial cells that cover the aorta. And at this point, these are not mature endothelial cells. All the system, all the cell types that we find in the embryo and are in constant development. And some of these cells will suffer an enormous transition that will go from this very long shape of endothelial cell into this round shape hematopoietic cell. And this process is what we now call the endothelial to hematopoietic transition, or EHT. Also that remembers the process of EMT that we link to the metastatic processes in cancer. So as you will see in some of the examples that we are studying, there are many of these processes, many of these signals that help us understand how cells are made, but also how cells behave, and also how parallel are some of these processes with what the cells are doing in a cancer, in a tumorigenic environment. So this is how it looks like in real life, and this is an image of a longitudinal section for an embryonic aorta, in which we can see the CD31 or PCAM, these endothelial cells in purple, and this is the light of the aorta, and this is the region of the embryonic aorta where all the vitelline artery is joining the aorta. We know in many places of this aorta we can detect around in the mouse around day 10, 11 of development, and in the human around week 4 or 5 of pregnancy, we can see these clusters, these groups of cells that have now changed this shape and now have transformed into what we know are hematopoietic cells, because they express many hematopoietic markers and transcription factors. Here we can detect them by the staining with c-KIT, and c-KIT is one of the receptors that will help us to identify the cells that have already made this transition from the endothelium into the blood development. What we have started to understand in the recent years are all the different cell types that compose these clusters. Because when we look at the staining, they look very homogeneous for some markers, but when as much as we keep getting more information and knowing what these cells express, we know that all these clusters of cells are heterogeneous. They contain different cell types within the hematopoietic lineage, but they are not all of them the same cell type. We know that a few of these cells are hematopoietic stem cells. And what we mean by hematopoietic stem cells are cells that when we transplant them in an irradiated mouse, these cells are able to recover the hematopoiesis of this animal. This is a very stringent assay that actually we are lucky in the hematopoietic system, because we can test if we think that we have a cell that has a characteristic of hematopoietic stem cell. We can directly test that in this assay that has been developed for more than 50 years and actually is the base of all the hematopoietic transplantations that leukemic patients have to go through when they have a compatible donor to treat many leukemias. So within using this assay in the mouse, we know that in one embryo at this stage of development, I say at day 10 or day 11, we have as little, or we can detect as little as 1 or 2 cells within the whole embryo that have these characteristics. And these cells are all residing within these clusters. So one of the main objectives of the lab has been trying to understand which cells are stem cells and how do they, what are the signals that is giving these few cells the characteristics to be stem cells. And why is this important? Well, the importance of this question resides mainly in one is basically to know the cell-cell communication and cell-cell interaction mechanisms that then can tell us how cells change shape, plasticity, and on the other hand, it has a useful objective, which is at some point we have been trying now for many years and we don't have the answer yet, but we really want to try to make these cells in the laboratory in vitro, because source of, we need hematopoietic stem cells to treat patients. We need cells that people can, we can transplant people with monogenetic diseases that may be replaced, the genes may be replaced in the hematopoietic system. And for this, we need compatible hematopoietic stem cells to be able to treat everybody, because very few people, in fact, have compatible donors if they need this kind of transplants. So that's one of the main reasons why we want to study this process. We want to understand it. And I want to spend a little bit of time to tell you how are we addressing these questions and the technical limitations that we have to study these specific tissues. As you may have guessed, there are the main limitation we have is that we have very few cells in each embryo, and we don't really know which ones are they. So we have a few clues, a few markers, but we don't really know which ones, which of these which of these groups of cells, which one is the one we're interested in. And try to understand how are these cells behaving and what are the cells that we want to study. We have put a lot of effort in trying to visualize how this is made. And the idea is that we would like to put a video camera within this aorta of the embryo at this time point, at this time of development when these cells are appearing, to be able to see them. But this is not that easy, because the aorta, even the embryo is inside the uterus, and then the embryo, once we take it out, is not transparent, and we need to create this transparency to be able to visualize with the microscopes that we have. And one approximation that we have been working on, especially this person, Cristina Porcheri, who was previously in the lab as a postdoc and now has her own position in the University of Zurich. And when Cristina came to the lab, she wanted to visualize what these cells were doing. And we need several tools. So only setting up this kind of experiments, it takes us quite a long time. We need tools that we can label the cells of interest. And we need tools that can label all the cells as well. Otherwise, we cannot localize them. So with this, we have different mouse transgenic lines, in which some are specific of the hematopoietic cells that are being generated at that time with these fluorescent proteins. So these cells will have colors that will express molecules that will be emitting different fluorescence under the excitation, the right excitation. And we have other mouse that have other molecules that basically will label the rest of the cells, so we can relatively place our cells of interest. In addition of having the right colors in the right cells, we can also inject antibodies. We can take the embryos out of the uterus and they will stay alive for some time. We can inject antibodies in the heart, and they will be distributed into the aorta. So we can label the cells in these tissues. And since we cannot image the whole embryo because we don't have the resolution to get into this internal part of the embryo, we need to chop these embryos. Basically, we make slices with these kind of choppers, and you can see here in this eye ring that we have the different embryo slices that we can embed in agarose, and then we can go and look in the microscope and actually record them and do time-lapse videos. So with this, we have been able to generate very nice images of the sections, of thick sections of these aortas. That here we have it labeled in red because this is the CD31 antibody that is labeling all the aorta. And we can see in yellow the hematopoietic cells because this is the CD41 YFP reporter, in which this fluorescent protein is expressed and goes to the membrane of all these hematopoietic cells, all these hematopoietic clusters that I was telling you that they form in these embryonic aortas. The rest of the cells are labeled with this histone GFP, and they are basically showing us the rest of the embryo architecture. And when we look at this, the interesting thing that we now can go and study is we can see these cells, we can follow them, we can actually see them dividing, like you can see here. So we can quantify, record and quantify many of these processes that they are doing in this during this time. So this has been a very interesting technique for us and has allowed us to go very deep into how are the cells behaving. And one of the first questions that Christina tried to answer was how these clusters, these hematopoietic clusters, were initiated for the first time in the aorta of the embryos. And I'm just showing a summary of many recordings in which she could see that there was one cell that was having these shape changes that basically was bulging from this, emerging from this endothelium. And this cell that was already labeled with our fluorescent protein, which we can see here, the first thing that was doing was dividing. So this was, we concluded that this is the first event that is happening when the clusters are formed. Cells emerge and divide. And so if cells emerge and divide, we assume that these will continue dividing to actually generate a whole cluster that will be clonal, and it will be coming from this original cell. And we actually developed quite sophisticated techniques to address this question, and one very visually very rewarding, but actually very technically very demanding, is the use of what we call the confetti mice. And these confetti mice, they have been used in different systems to actually understand how the tissues are made and are organized, and how what's the hierarchy of the cells that compose the tissues. So what is the confetti mouse? The confetti mouse are these transgenics that they carry this type of genes, which this cassette contains different fluorescent proteins And all these fluorescent proteins are surrounded by these LOXP sites, which are recombination sequences that when they are exposed to a specific recombinase, they will stochastically recombine and form one expression cassette that will give rise to one specific color. If we use this tool together with endothelium specific recombinase, which is we can do with this CRE recombinase that we control with tamoxifen and we control with the cadherin, which is a very specific endothelial gene. We can have recombination of specific cells in this aortic endothelium in the embryo. And as you can see, the cells is this, we set up this technique so this recombination is happening not in all the cells, but in very few cells, very sparse cells of the endothelium. Why do we want to do that? So we want to do that because we want to know whether this single cell that we know emerges from the endothelium and divides, keeps dividing until forms a monoclonal cluster, and within this cluster hematopoietic stem cells develop. Or on the other hand, there's several cells from the endothelium that join in and actually make these polyclonal clusters, and their cells coming from different origins. So to address this, we basically, Christina analyzed many of the embryos that had recombined the endothelium, or specific cells in the endothelium, and analyzed what happened with those clusters of cells. And here we can see that these hematopoietic cells that express c-KIT, they now, when we are lucky, not all of them, but when we are lucky, cells express or contain this one of the 4 fluorescent proteins. And these, what we could see is actually the summarized in this graph. And as we expected and as we had seen with our videos, all the clusters that were 2 cells, that had 2 cells, they were and were labeled, they were labeled with the same color. Also, the clusters that had 3 cells, they were labeled with the same color. So in this case, this was confirming that first, the very first thing that these cells do, they divide, and the very early stages, the very small clusters, they are monoclonal. However, as soon as these clusters reach 4 or 5 cells, what we can see is that there is other either non-labeled or cells labeled with other colors that actually are participating in this cluster. So for us, this was a clear result that was telling us that first cells start dividing, but as soon as the clusters reach a specific size, then other cells from the endothelium join into the cluster. So we concluded that the clusters were monoclonal, but started monoclonal, but then when they grow, they turned polyclonal. And what else? This is just an example of the techniques that we are using to understand how these cells behave and are made. But in fact, what we are really interested is in knowing the signals that control these processes and this behavior. And we know in fact, most of the developmental tissues are controlled by a very restricted number of signaling pathways that actually repeat or keep repeating their functions in many different tissues. And these signals also are crucial in any of the pathogenic processes that happen. And obviously, in our case, we are very interested because they are, most of them, crucial signals that get deregulated or mutated in cancer. For example, this is just a review that I wrote a couple of years ago with actually one of the people, one of the representative scientists in this field, who's Elaine Zerzak. And we made this summary just to represent all these pathways in which, for example, we can recognize TNF and NF-kappa B, or TGF-beta BMP with SMADs, Wnt, beta-actinin, and of course the one we're going to talk about in the next few minutes is Notch. But as you know, all these pathways actually regulate many different processes, and they are very important in understanding what the cancer processes are like. So, as I said, we will we in our lab have been very interested in this specific pathway. This is the Notch pathway, and this is one of the models that we use to study how cells interpret Notch signal and why cells need this Notch signaling to develop and to remain as a healthy cell. And we know this is when I'm going to actually I just want to take a very few minutes to say that Notch actually contributes to many different stem cells, and we wrote this review with Dr. Luis Espinosa many years ago, but this is basically still true for all of it, and there is only been increasing the number of tissues and the number of cell types in which we know that Notch is crucial. In the system that we are looking at, we know that Notch is required to make this hematopoietic stem cell development, and we have been trying to understand which elements of all this pathway are important to make these cells. We know that this is a very basic cartoon of how Notch works, and Notch is a cell-cell communication mechanism that the cells use basically to decide what to do. And in general, the interaction comes from one ligand that is expressed in a neighboring cell that interacts with the receptor that is Notch. And when this interaction happens, the receptor is cleaved and the intracellular domain of Notch goes into the nucleus and activates transcription of different target genes that in any different system, it can have its own context-specific genes. So to study how this system works, basically what we are, or the technology that we have been using for many years now is to isolate this part of these tissues in the embryo and then ask the question whether there are stem cells in these tissues if we manipulate or if we delete one of the key members of the Notch signaling pathway? And the answer is that for many of these, many of the members of the family, we have a very clear phenotype, which is there is no stem cells. And this happens for Notch1 and all these ligand Jaggedl1 and also some of the downstream targets. There's also other different receptors and ligands that are still under study or that we have not found any function for them. But basically, the idea is we take these AGMs from wild type and from knockout mice, we put them in irradiated mice, and then we check whether we can see the donor cells that have recovered the hematopoietic system of this mouse. And this is what we see in the wild type, but this is what we see in the knockout. There are no cells, if they are lacking one of these molecules, that can contribute to this hematopoietic system. And another of the important things is that Notch, it's not only important to make hematopoietic stem cells, as I told you, but it's crucial to make these arterial cells in the endothelium. So these are two processes that are very closely linked because, as I told you, the hematopoietic cells originate in the aorta of the embryo. So the arteriogenesis is also happening at the same time. And cells have to interpret the Notch signals, and the expression of the ligands will be crucial to understand when they go into making hematopoietic cells or into making the arterial cells. And this is something that we have been studying also for many years to understand what's the interplay of the ligands, why we have one important receptor in both processes, in the hematopoietic development and in the arterial development, and they both depend clearly on Notch1, but we are just starting to understand how this receptor interprets the signals coming from different ligands. In this case, Jagged1 for the hematopoietic program, and delta-4 would be in the arterial program. We know quite a lot about the ligands and how they interact with the receptor at this point, and we know that the signals that we get downstream of these ligands are also can be different and can have a different kind of Notch activity and a Notch response. And this is what we have been trying to keep working in the last years. And basically, with this, we were very lucky to have tools to now understand what one of the other ligands that had not been explored, that's the delta-4 ligand, was doing in these hematopoietic clusters where it is expressed. And I'm just going to go very briefly in this. This is a more detailed study that we have done and we have just published, but just to illustrate how we knew how we can understand what these different ligands are doing. In this case, we already knew that Jagged1 was very important to make hematopoietic stem cells, but we didn't know where delta-4 was important. And considering that delta-4 has a very strong effect on the artery, that was difficult to study. However, we came with, we could use this very useful tool with our blocking antibodies developed against specific ligands or receptors of the Notch family. And this has been crucial to actually go and inject these antibodies and then look at the effects that it would have in this developing aorta. This is different than studying knockouts, because for generating knockout animals or conditional knockouts, we need much more time than we need when we use these blocking antibodies, that we can go very fast. We can inject them and study these embryos, study the effect in 2 or 3 hours. And this is what Christina did. She injected these antibodies, and I'm just going to summarize what she saw in only incubating these embryos for 5 hours with the blocking antibody. So if we block delta 4, we can see we have controls that have the normal size clusters. In general, they are pretty small clusters. Most of them are down here in 5 to 10 cells. However, when we block with delta 4s in a very quick way, suddenly what we see is these huge clusters, some of them getting to double and tripling the size of the normal cluster. And we have been, of course, very interested in why this was happening. And basically, we have more number of c-kit cells in the clusters, but most all the it's like all the all the clusters suddenly start to grow, but we don't have more number of clusters. So the number of clusters that we can count in the whole embryo remain the same. The number of c-kit cells in the clusters are much more. So they are bigger. We have bigger clusters. And we know it's not proliferation. We have done all different experiments. It's also a very quick process. And what we have also explored is whether there is more incorporation of cells into the cluster. And I'm not going to go in detail in these experiments because I'm running out of time. But basically, with this confetti mouse, we could see how many more recombinant cells were included into the clusters. And suddenly, we had clusters that had incorporated many more cells. And in fact, we also could see by these time-lapse videos how neighboring cells in the clusters were suddenly being incorporated within the cluster unit, indicating that these clusters are growing by recruiting more cells. And just very briefly, I want to also tell you that we know the type of cells that are being recruited into the clusters. These are these GFI1 cells that we can now label by transgenic reporter, and we know they are hemogenic cells, so the cells that are still in the endothelium have started to be are starting to show some hematopoietic traits, but they are still not cluster or c-kit positive cells. So finally, what we have seen is what this process is doing is just making the hematopoietic cells in this embryonic aorta, making them better for engraftment and to have a more robust transplantation in the mouse when we actually use cells that have been treated with this delta 4 antibody and put them in the transplantation assay. So just as a summary, what this just told you is that it is a specific notch signal that is delta 4 is actually keeping cells in the endothelium and limiting the number of cells that actually are going to become hematopoietic stem cells. As soon as we block this signal, what this is like, this control is released, and then many other cells can go into the cluster. And obviously the cells that go into the cluster seem to be the right cells, because what we can then see is that we have more hematopoietic stem cells that are being formed. So we think that understanding all these signals is crucial to actually understand how we can make hematopoietic stem cells in vitro, on how we can make better hematopoietic stem cells that have the capacity to engraft into immunodeficient organisms, and this could help in the future to obtain these cells. Just to finish, I want to thank all the lab members. I've actually all these former members that have been directly involved in this work are here. There's many other former members who were not involved that are not here, but I just mentioned Christina's work, and also Roshana Tambiraja, who is taking over and continuing to study all these notch signals in the embryo. Yolanda, Jessica, Luis, Arnau, and the rest of the people. Many thanks to Luis Espinosa and his lab because of also their intellectual contribution to all the projects, and rest of collaborators for this work. So I will just thank you for your attention and take questions. I guess I pass it to Vanessa.
Thank you very much, Ana. Amazing presentation. I'm going to switch to Spanish just to do a clarification. Lo siento porque no he explicado al principio de la sesión que cualquiera que quiera hacer alguna pregunta puede irlas registrando en el apartado de preguntas. Tenemos alguna de participantes que ya sabían cómo funcionaba el sistema, pero imagino que otros pues no estaban al tanto. Así que los que quieran pueden ir escribiéndolas. Sorry, we will continue in English from now. So if that is okay, Ana, I'm gonna proceed to read some of the questions we have here. Cristina Viera is asking, well, she's saying thank you all for the great initiative and for the talk. And she would like to ask if you know if the HET process have been described in tumor context, and if also the inverse process, HSC becoming endothelial, has been described in tumor.
Okay, yeah, that's a really great question. We have not been able to see or to describe, or as far as I know, anybody has been able to describe a similar process in tumors for EHT, for the endothelial to hematopoietic transition. It can be certainly, I think it is a process that may be able to happen because there has been reports from the group of Rafi Shahins in which they take adult endothelium, actually from the lungs, and reprogram it into hematopoietic stem cells. And this is something that certainly can be done in the laboratory. We don't know if this happens in vivo. As far as we know, we don't know. And about the other question, which is the reverse process, I just see it into endothelium. This is again there are no reports in which HSC can become endothelial cells, as far as I know. For many years, it was thought that there would be these bipotent cells where they would be able to do both, but I don't think this has been, I mean, most of the data that we have been generating or people has generated in the recent years goes into the direction that the process is irreversible, at least in a physiological way. There may be, obviously, the plasticity of cells is something that we can engineer and probably change, but in a physiological way, it's not known to happen.
Okay, thank you very much, Anna. Another question, thinking about transplants, how immunogenic are stem cells? And a second question, does HLA matching matters much in this context?
HLA, yeah. Yes, it does matter. Probably, I don't know if it matters for the hematopoietic stem cells, probably it does matter, but then these hematopoietic stem cells will generate mature cells, which in the case that even if the hematopoietic stem cells were not that immunogenic, the other cells will be. We need to do, when we do the transplants, so the mouse is the perfect system because we use for all these experiments compatible strains of syngenic mice. For thinking in a human application, this is obviously this is something that we will need to address in the future. One possibility that obviously solves this problem is generating, or in an ideal world where we can generate iPS from the patient and then differentiate these induced pluripotent stem cells into hematopoietic stem cells of the same patient. So we for sure don't have the problem of the immunogenicity.
Thank you. A question from Joaquin Arribas. What are the differences between human embryonic hematopoietic stem cells and adult stem cells?
So there are differences. There are many differences because we know that these cells keep evolving, and there are in a phenotypic way we already know some of the receptors in the membranes that are not exactly the same. So the embryonic cells can do the job. Let's say the job is they can reconstitute the hematopoiesis of an adult organism, but they need to mature probably. And we think this is happening in the fetal liver. In the fetal liver, they are acquiring more characteristics of an adult stem cell. And this probably has to do with the response of these, how these adult stem cells can respond to the different conditions, different stress. And so it's obviously our test for identifying these cells is we want to have the cells that can reconstitute an adult mice, but there are many cells that cannot do that robustly in the embryo, and probably we have they still have some way to go before they become the real cells.
Okay, another question from Ruben Sebastian Pérez. He said, nice talk. You have shown how hematopoietic cluster rise from AGM regions at E10.5 stage. I'm curious to know your thoughts about any extraembryonic contribution, maybe yolk sac or even placenta at earlier development stage. Can you develop on that?
Yeah, this is, yeah, we could be discussing for hours. In fact, we have in our field, we have been discussing for years about this specific question. And what I think the contribution is important because there are many cells being produced in extraembryonic tissues, not only in the yolk sac, also in the placenta. I think that the main site for the early events of hematopoietic stem cells is it's within the embryo, but the cells that are produced in the extraembryonic tissues probably help a lot in making the right microenvironment. Maybe not in the AGM, I'm not sure about this, maybe more in the fetal liver, because in fact, when stem cells arrive in the fetal liver, it's full of other hematopoietic cells that have already come from the yolk sac, for example. So I think all these cells are contributing to the final job, but I am not sure that they are making stem cells per se in these other tissues. But of course, this is still one of the questions that is very actively being investigated. And the problem comes when we deal with embryos, very small numbers of cells, very small organisms, and then the circulation between the intraembryonic and extraembryonic tissues makes it very difficult to establish very strong conclusions.
Thank you, Anna. We are running a bit out of time, but questions keep coming, and I don't want to leave any of them. So Tony Celia says, the AGM cluster are polyclonal based on your study. But what about when they go to the fetal liver and then to the bone marrow in adults? Are they still polyclonal, or some clones are more competitive to form the deriving the hematopoietic cells? Yeah, Anna, can you hear me?
Yes, yeah. I was trying to think the answer. What I think is that most of the cells in these clusters, they will not be the final hematopoietic stem cells. So they are helping, or they are the stem cell microenvironment at that time, and probably they can go to the fetal liver. I don't know how longer they stay around, or whether they basically are more progenitors that they will be extinct. There's some clonal analysis of adult hematopoiesis, and it seems that there's probably hundreds, at least in the mouse, of original cells that then they will give rise to the whole clonal hematopoietic system, or to the hematopoietic system.
Okay, thank you, Anna. And the last question from Raúl Peña. He said, nice talk, as always. He's curious about if the small monoclonal cluster of the two free cells induce neighbor cells to differentiate and to be recruited inside those clusters, or just recruit other small monoclonal clusters that emerge independently, but close to them.
So I couldn't go into the details, but I tell you it's not a merging of clusters what happens. So it's not a recruitment of other small clusters. Or at least this is what we think. We have done mathematical modeling with all this data that we have with the help of David Sprinzak in Tel Aviv University. They have done all the mathematical modeling in considering both conditions. One, that there is a single cells that are recruited, and another that there are clusters that can be merged, and the model fits with that there is single cells that are being recruited or individual cells that are being recruited into the clusters. Although we have some very big clusters in our data that we cannot exclude that they are coming from the merging, but we don't think it's the general process of what we're seeing when we block delta 4.
Perfect. Thank you very much, Ana. Let me just switch to Spanish again, just to close the session. And solo daros las gracias de nuevo por haber participado en la sesión de hoy y darte las gracias, Ana, por haber presentado este espectacular trabajo. Y quería aprovechar la oportunidad también para felicitarte por tu nueva posición como directora científica de Ciberón y para enviar un abrazo desde aquí a todo el colectivo de Ciberón, que ya sabe que para mí es un colectivo muy importante y estoy segura que ahora en tus manos van a tener muchos logros colaborativos. Para todos los demás, simplemente deciros... Para todos los demás, simplemente deciros que continuaremos la semana que viene con nuevos seminarios y que tendréis toda la información en el canal de YouTube del IMIM y en la página web del IMIM. Muchas gracias, Ana. Muchas gracias a todos.
Muchas gracias.
Chao.